Hello everyone, this is Dr. P. Mary Anupama, Assistant Professor and Head of the Department of Biochemistry, St. Joseph's College for Women, Visakhapatnam. So today we'll be studying the topic Glycogenolysis. As a part of the presentation, the topics that will be covered are basic introduction to what is glycogenolysis, followed by the location or the site where it takes place, and then the steps of glycogenolysis in detail, and followed by regulation of glycogenolysis. What is glycogenolysis? As such, it is a metabolic process where the glycogen which is stored in the body, either in the liver or in the muscles, it is converted to glucose. That means the glycogen is broken down to glucose. So this is what is glycogenolysis. So this process, actually it is a very reliable source or the pathway through which the energy, it is supplied to the body in the form of ATP, and it also helps us to maintain the blood glucose levels, mainly when a person is going through strenuous exercise or when fasting or doing some works where high amount of glucose is required by the body. So glycogenolysis is nothing but breaking down of glycogen to formation of glucose. The sites where glycogenolysis takes place, these are mainly two tissues, that is the liver and the skeletal muscles, and primarily the reactions occur in the cytosol of these cells. Either it could be liver or skeletal muscles. So glycogenolysis in the liver. So in the liver, glycogenolysis occurs mainly to maintain our blood glucose levels when a person is fasting or starvation or in between the meals. The liver, it contains this enzyme glucose 6-phosphatase, which converts glucose 6-phosphate into free glucose. So this allows release of glucose into the bloodstream. And this is very important to maintain homeostasis of blood glucose levels so that the energy could be supplied to those tissues which are glucose dependent, like we have brain and RBC. So glucose 6-phosphate is converted to glucose by glucose 6-phosphatase. Glycogenolysis in muscle tissues, skeletal muscle tissues. This provides instant energy, mainly during muscle contraction or when a person is performing physical activity. Here, these skeletal muscle cells, they lack glucose 6-phosphatase. So, the glucose 6-phosphate which is produced, that will enter glycolysis and it will start providing the ATP which is required for the functioning of the muscles. Then glycogenolysis in muscle is only for intracellular energy use and not for maintaining blood glucose levels, as you see in the case of liver. Subcellular location. So many enzymatic steps of this glycogenolysis, they occur in the cytosol of the cell. Hence, it is a cytoplasmic pathway. However, the regulation of glycogenolysis, it is under hormonal control, and that interaction is made through membrane receptors, as well as we have some cytosolic secondary messengers like cyclic AMP and calcium which is involved. Apart from the liver skeletal muscles, there are also some minor sites like you have tissues like the kidney, where limited activity of glucose 6-phosphatase is there, astrocytes in the brain, etc. Now let us see the steps of glycogenolysis. The first step, it is glycogen phosphorylase reaction. Glycogenolysis is initiated by the enzyme glycogen phosphorylase. It acts on the non-reducing ends of glycogen molecules. This enzyme, it catalyzes the phosphorylated cleavage of alpha-1,4 glycosidic bonds, thereby releasing glucose 1-phosphate. This reaction, it uses inorganic phosphate instead of water, and it also requires pyridoxal phosphate as the coenzyme. So here you have, say for example, this is the glycogen molecule. One glucose molecule is released as glucose 1-phosphate, leaving a glycogen which is N-1. This is the first step. Next comes the role of debranching enzyme. Here at the branch points, this enzyme comes into role. It is a bifunctional enzyme in its activity. It has a transferase activity as well as 1,6-glucosidase activity. Coming to the transferase activity, it is mainly involved in the transfer of 3 glucose residues from the limit branch to a nearby linear chain. While the alpha-1,6-glucosidase activity, it is involved in the hydrolysis of the remaining alpha-1,6 linked glucose, releasing it as a free glucose, which is not phosphorylated. So at the branching point, where you have the limit dextrins, the debranching enzyme releases free glucose by cleaving alpha-1,6 bond. Thereby, you will be getting linear glycogen molecules which are available for another round of hydrolysis. The third important step is the conversion of glucose 1-phosphate which is formed to glucose 6-phosphate. So the glucose 1-phosphate which is formed, which is released in the first step, it has to be converted to glucose 6-phosphate, and this is done by the enzyme phosphoglucomutase. This enzyme, it works via a transient state where you have glucose 1,6 bisphosphate as the intermediate which is formed. So the glucose 6-phosphate, it is the key branch point in metabolism. In muscles, it enters glycolysis. In the liver, it is converted to free glucose, and then it enters into the blood to maintain homeostasis of blood glucose levels. Fourth step is the conversion of glucose 6-phosphate to free glucose in the liver and kidney only. We are not talking about the muscle. So here the muscle lacks this enzyme glucose 6-phosphatase while it is present in the liver and kidney. And this one hydrolyzes glucose 6-phosphate, releasing glucose and inorganic phosphate. So this free glucose which is produced here, it will enter the bloodstream to maintain homeostasis. While muscle cells, since they lack this, what happens to the glucose 6-phosphate is, it enters glycolysis. Rather than contributing to blood glucose, it enters glycolysis, and through the glycolysis, the glucose is hydrolyzed, meeting the energy needs of the muscle cells. So glycogen phosphorylase, that is the enzyme which is releasing glucose 1-phosphate. Then we have the debranching enzyme which is removing the branches and releasing free energy. Then we have phosphoglucomutase which is converting glucose 1-phosphate to glucose 6-phosphate. And finally the glucose 6-phosphate, it is converted to free glucose. This is how the glycogenolysis is taking place, resulting in the release of glucose. This is the oral pathway of glycogenolysis. Glycogen, which has glucose moieties linked by 1,4 and 1,6 linkages. Glycogen phosphorylase initially acts releasing glucose 1-phosphate moieties. Then the glycogen phosphorylase activity continues until some extent wherein it stops here at the branch point. So transferase enzyme, it transfers a trisaccharide unit to another branch point. So once this is transferred from here to here, the debranching enzyme comes and it cleaves the alpha-1,6 linkage here, and the glucose moiety is removed here. Glycogen phosphorylase further acts releasing glucose 1,6 phosphate units. So after the series of hydrolysis, glucose 1,6 phosphate is converted to glucose 6 phosphate and then to glucose. This is the oral pathway. Now let us understand regulation of glycogenolysis. Glycogenolysis is nothing but the breakdown of glycogen into glucose, and this is a very highly regulated process, and this ensures that glucose is released according to the body need for energy. And this regulation, it involves allosteric control, hormonal regulation, and colon modification of enzymes. First one is key regulatory enzymes. Mainly the glycogen phosphorylase, that is the rate limiting enzyme in glycogenolysis, and it exists in two interconvertible forms. We have phosphorylase A, which is the active form, and it is the phosphorylated form, while we also have the inactive form phosphorylase B. Okay, it is the dephosphorylated form. Interconversion of these two will be taking place. So, this glycogen phosphorylase enzyme, its activity is controlled by phosphorylation and allosteric modulators, which vary depending upon the tissue, like in the liver or in the muscle. Second regulation is, we call it the hormonal regulation. Yes, hormones, they regulate glycogenolysis through signal transduction pathways that modify the activity of glycogen phosphorylase. Again, the hormone regulation, it could be studied in the liver as well as in the muscle. First is in the liver. So in the liver, you have this glucagon and epinephrine. Okay, these glucagon mainly during fasting, and epinephrine when you are in stress or during exercise. These are the key activators. And these hormones, they bind to the receptors and they activate adenylate cyclase, which increases the cyclic AMP levels. So the cyclic AMP in turn activates protein kinase A, which phosphorylates phosphorylase kinase enzyme. The phosphorylase kinase, which is formed here, the phosphorylase kinase further phosphorylates, okay, glycogen phosphorylase B, which is the inactive form, to phosphorylase A, which is the active form. So these are the sequence of things which take place. So once again, glycogen or epinephrine, they activate cyclic AMP formation. Next, protein kinase A. Next, phosphorylase kinase, which in turn phosphorylates the glycogen phosphorylase B into its active form, glycogen phosphorylase A. Hormonal regulation in muscle. So in the muscle, it is epinephrine. It is the main hormone which is activating via the cyclic AMP pathway. So additionally, muscle contraction, it increases intracellular calcium, which activates the phosphorylase kinase through calmodulin. So the AMP, which is produced during ATP hydrolysis, also directly activates the phosphorylase B. So you have epinephrine activation, which triggers the cyclic AMP pathway. Then this cyclic AMP pathway, additionally, it increases the intracellular calcium, which is by muscle contraction. Then this activates phosphorylase kinase through the calmodulin. Calcium binds to the calmodulin, which activates phosphorylase kinase. Now the phosphorylase kinase in turn activates glycogen phosphorylase. Now this glycogen phosphorylase, when it is phosphorylated, the glycogen phosphorylase B gets converted to glycogen phosphorylase A. So it is an allosteric regulation here again. Third way of regulation is allosteric regulation. So this is seen in liver as well as in the muscle. So in liver, the glucose It acts as an allosteric inhibitor of glycogen phosphorylase A, promoting its conversion to its inactive form, that is glycogen phosphorylase B, when glucose levels are very high. When coming to the muscles, when this AMP mainly, it activates glycogen phosphorylase B, ATP and glucose phosphate. They will be inhibiting it, reflecting that adequate energy is there. ATP and glucose 6-phosphate, if they are there, it indicates that adequate energy is there. Thereby, they will be inhibiting the enzyme activity. We also have one more method, fourth one, inactivation. It is insulin-mediated enzyme inactivation by dephosphorylation. So when you have energy or glucose levels, if they are high, then insulin that promotes phosphorylation of the enzymes. So insulin, it triggers dephosphorylation of enzymes. We have protein phosphatase 1, which is activated to dephosphorylate the enzymes. Then phosphorylase kinase, which usually activates the enzymes, it is converted into its inactive form. That is, it is inactivated. Then glycogen phosphorylase. Okay, it is converted into its inactive form, the B form. Then glycogenolysis, it is inhibited. So if glycogenolysis is inhibited, then glucose levels will be reducing. That means glucose production will not be there. So glucose production will be reducing. Glycogenolysis is promoted. Glycogenesis is promoted, which means since glucose is more, then it increases storage. That is, glycogen synthesis will be promoted. So glycogenesis is promoted. So here you see that it is inhibiting glycogenolysis and it is promoting glycogenesis. That is how insulin acts. So in this topic, we have seen thoroughly the introduction where glycogenolysis takes place, steps involved, and then its regulation at various levels. The textbooks that are referred during preparation of the material include The Principles of Biochemistry by Linenger, Biochemistry by U. Satyanarayana, then Harper's Biochemistry. Thank you.